Hello, I'm Rogelio Arenas, and I'm gonna talk to you throughout all of my semi-structured interview. That's for my evidence too for molecular bases. Okay, so for my first question, I have what are the molecular bases that explain the advantages of the shark ignore antibody over common immunoglobulins? And first, well, the shark ignore is the immunoglobulin new antigen receptor. These are antibodies that are structurally distinct from the typical mammalian immunoglobulins. And then the advantages that this could have is, for example, the single domain structure, because they are composed by only heavy chains. Sorry, and they don't have like the light chains that we have, making them really small and more a lot more stable. Another one is their high solubility and their thermal stability, because they have like unique disulfide bridges, and they have like a hydrophobic packaging that they remain functional under the really hard conditions. Another thing is like they're really hard or tough like tissue penetration, because their smaller size helps them for a better like insert into the different tissues or tumors or something like that could be bad. And the another advantage that I learned of the ignore is that they have like unique antigen bindings that are called the BNR, because they recognize like recessed epitopes that are often inaccessible for like conventional antibodies. And the last one is that they have like reduced immunogenicity potential, because their sequence can be like humanized, like easier compared to the full size like antibodies. So yeah, that's why. Okay, so for my second question, I have what molecular and digital tools would I use for to design the synthetic like production strategy of the ignore in yeast? And well, first of all, like designing the synthetic production for the ignore antibody in yeast system, it's like really difficult and needs a lot of different techniques and digital tools as well, so we can ensure the efficient expression, the correct folding, and successful secretion of the antibody. For example, the codon optimization, because since the ignorant genes come from the sharks, its codon usage can be adapted, needs to be adapted for the optimal translation in the yeast, like the PK pasteurizers that we used, and also the expression vectors, because we have to. This is very important because we need to select the appropriate vector, like the one we chose, because they need to have the MCS, a strong regulated promoter, a secretion signal, and selectable markers. We also have to add the tags such as the his tag or something like that for the protein purification, and we can detect it like by affinity chromatography or something like that. As well as we have to choose our yeast host strain, so it can be like really compatible with the everything that we're using for the correct expression, as I mentioned, supporting our high yield protein production and as well as the secretion. And for the digital tools that I mentioned, we could use the SnapGene or BenchLink, so we can simulate the restriction cloning, the design of the primers, so we can check like everything and every detail that we need before like getting into the experimental part. And and yes, that's those are the tools that I will use. Okay, for my third question, I have what is the proposed theoretical strategy for cloning and the expression of the IGNAR gene in the yeast? So first, we have the codon optimization, as I mentioned, of the IGNAR gene for the yeast expression. Then we have to do a PCR amplification of the gene that we are targeting, and then we have to clone it into the expression vector. We have to choose the vector. We have to insert the gene in the MCS with the correct restriction enzymes, and as well as adding the alpha factors so we can secrete correctly. Then we have to transform it in the yeast. We have to first linearize the plasmid so we can have everything perfect. We have to use then electroporation, because that's the one we knew really well for the yeast transformation, as well as making the cells competent for all of these, first of all. And then we have, after having the electroporation and the transformation done in the plates, then we have to do the selection screening After the colonies grew in the selective media, we have to screen them, and we have to visualize if the gene we're targeting is really inserted in the plasmid. So that's why we use the colony PCR and well the sequencing as the end, and well we have to use the galactose or methanol depending on the on the vector we used, so we can induce the expression, and yes, that's our like strategy. Okay, so for my fourth and last question, I got how could I corroborate that the new ignorant antibody is being produced correctly, and that it's biologically active? So first, we can test like the binding capacity of the ignorant, while using the LISA sandwich LISA specifically. So we can have like a positive binding confirming like the functional antigen recognition that is here like in the picture. We can see how it works, and yeah, we can like make analysis for the kinetic parameters like the affinity constant, dissociation rate, everything that's that to confirm like the proper folding, and as well as well with the proper folding we have the expressed protein, the expressed antigen, sorry, and the interaction strength. And well, that's my interview in a different way. And thank you very much, Dr. CJ, for allowing me to do this presentation and this interview, and for you to accept it. And I hope you have a great day. And thank you so much for teaching me again in two different classes. I learned a lot, and I hope to see you soon. Thank you.